Hello, my name is Alexey Mishin. I'm from Moscow Institute of Physics and Technology, and today my talk is dedicated to the case study of structure-based drug design with Cysteinyl Leukotriene G-protein coupled receptors. So I work in the Research Center for Molecular Mechanism of Aging and Age-related Diseases at MIPT. It's a big research center with 10 laboratories, 9 advanced research platforms, and we also have a chair of biophysics which also has 2 master degree programs dedicated to different aspects of molecular biophysics and structural biology. Our center possesses quite a strong publication activity, and we also work in a big network of international collaborations with academic institutions all over the world, and also with some R&D departments of pharma companies. And now, the international focus is shifted to mostly to China and South Korea, for instance. And our main research focus are membrane proteins, which are major functional elements of cells. If speaking of membrane proteins, they are very important drug targets, and if we see the market data and all the drugs, the share of drugs which target membrane proteins is quite huge, and there are several major types of membrane proteins which are in focus. The first one is G-protein coupled receptors, then ion channels, enzymes, transporters, nuclear receptors, kinases, and other. And if we will see on the statistic of very top pharmaceuticals by retail sales, we will see that 60% of bestsellers drugs target membrane proteins. The research focus of my laboratory are the G-protein coupled receptors. G-protein coupled receptors, they transmit signal from outside the cells, and when they are activated, they transduce a signal inside the cell and cause a lot of types of extracellular response, modulating gene expression and a lot of intercellular processes on genome level, and also they regulate activity of other proteins and protein complexes. GPCRs are activated by ligands which possess different types of chemical nature. They could be ions, biogenic amines, amino acids, lipids, peptides, and proteins, and also small molecules of different kinds that can be found in human metabolome. And GPCRs also themselves are very important drug targets, and if we will look at the drug discovery pipeline, when you are working with GPCRs and many other targets, it is often important to implement the strategy of structure-based drug design. And structure-based drug design makes the process of drug discovery quicker and cheaper, and in many cases the effect is very significant. And protein structure can be essential to at the stages when you identify the hits and then optimize them to leads compounds, and then you can use the structure to find better compounds without extensive screening. How we do it at MIPT? We have research platforms where we can study our proteins from genes to structure. First, we screen a lot of gene constructs and we express them in eukaryotic expression systems, mostly in insect cells. So we use SF9 insect cells expressions, and we screen the existing ligands to find those ligands that stabilize the receptor. Then we purify the protein and crystallize them and go to synchrotron radiation sources or X-ray free electron lasers to get the crystal structure. And also nowadays we are also using cryo-electron microscopy, but for the objects that I will show you today, the structure was obtained by X-ray crystallography on the synchrotron sources and X-ray free electron lasers. So everything before the and including crystal growth we do at MIPT, and then we go to the powerful X-ray sources. Unfortunately, in Russia we don't have very bright radiation sources, only the SCIF synchrotron in Novosibirsk that is currently under construction. We hope that in future it will be modern generation synchrotron source and we will use it. But currently we need to go to Shanghai or South Korea, and also now it's possible for us to still to go to Trieste synchrotron and study our crystals. So there are several approaches for the crystallography that we use. It is classical X-ray crystallography on a single crystal, so where we mount the crystal on a loop and then it is exposed to X-ray radiation, and then we see the diffraction pattern and solve the structure. But if the proteins, protein crystals are very small and fragile, we use so-called small wedge crystallography when we obtain only partial diffraction data set from each crystal, and then we combine data coming from different crystals, and then we have the whole diffraction data set from many samples. And when the protein target is very challenging, we use so-called X-ray serial femtosecond crystallography, which is implemented on X-ray free electron laser facilities, which are the most bright sources of X-ray radiation, and they use the principles which is called diffraction before destruction. And when the crystal is exposed, you can to the radiation, you can obtain only one diffraction pattern from a single crystal, and then it destroys completely into a plasma state, but you can obtain the pattern before it destroys. But you should have thousands of crystals, because one crystal gives only one pattern, and you need to get the whole diffraction data set. And yeah, this approach is very beneficial for drug discovery, and you can read the details in our recent review. And also what is nice that you can use crystals at room temperature, and you don't have the artifacts in structures that can be made by cryocooling, and you don't have you have no need to harvest the crystals. You just use the suspension of crystals or crystals dissolved in so-called lipidic cubic phase, and so on. and there are only a few hard X-ray free rectal lasers in the world available: European XFEL, Stanford XFEL, Swiss XFEL, and there's also in PAL in South Korea and SACLA in Japan. And so we used to obtain data in this at European PAL and LCLS in Stanford, and now only PAL is available for Russian scientists. But in a couple of years, there will be in Shanghai, there will be the most powerful on board free rectal laser called Shine Facility, and we a couple months ago visited the construction site there. So, and the X-ray free rectal lasers are very huge, nice mega science facilities, and they help structural biologists a lot in different projects with challenging targets. But the procedure of crystal growth is not so classical in case of preparation for X-ray free rectal lasers research, because in normal crystallization you use crystallization plates, but here you should crystallize your samples in syringes and transfer then your samples to special injectors that expose your sample to ultra bright X-ray beam, and you should grow a suspension of tiny micrometer or 10 micron length crystal, and you should have thousands of them. And that is often made by use of so-called lipidic cubic phase, and that helps to inject the crystals into the beam. And as in case of membrane proteins, you grow the crystals in such lipidic cubic phase. And also there is a special pipeline of data processing that is more complicated than in classic crystallography, because you should integrate all the patterns obtained from different crystals, and the data treatment algorithm are quite complicated, and special software is currently developed to solve these problems. As for our targets, we're working on, and we currently also continue working on Cysteinyl Leukotriene receptors. These are very important targets for treatment of asthma and inflammation diseases of broad other spectrum, like allergic rhinitis, urticaria. But asthma, is the strongest burden on society. There's a impressive statistic in the United States. In Russia, it is also a very difficult disease. 10% of children suffer from asthma in Russia, but there is a problem that current therapies, for example, the felocast, montelukast, pranolocast, which target only the first type of Cysteinyl Leukotriene receptor, it is not proper treatment for about 30% of patients who are not responding to the therapy. And we are working on development of a compound that can we started it to do it with in collaboration with Japanese owner pharmaceutical company, and we want to develop using structure-based drug design compounds that target both Cysteinyl Leukotriene receptor type 1 and 2, and which such compounds should be available as oral treatment. for patients. And we grow crystals of these receptors in complex with different available ligands, and we solved structure, published it in a couple articles. One was in Nature Communications, another was in Science Advances, and this structure allowed us to understand molecular determinants of ligand specificity, and also gave an insights how to design compound which target both types of the receptors, and yeah, it was the first structure of this receptors that was obtained. And then we made virtual ligand screening with compounds from Yenamin library, and found 5 compounds with affinities that were better than 1 micromole. That's not very great, but yeah, we now also continue this work, but nevertheless there's a they were found through the screening with micromolar affinities and quite nice characteristics. But then we found that there is a single point mutation, L129Q, which is associated with orphan cancer disease called uveal melanoma, and one of these compounds showed activity against this pathogenic form of the receptor, and there is no treatment of this type of cancer. Only on the surgery can solve this problem. But we want to make a compounds that can be a therapy against uveal melanoma, and this mutation is a proven oncodriver mutations in about 3% of all cases of uveal melanoma. Yeah, it's an orphan case, and but nevertheless, it can help patients who has these mutations to treat the cancer, and it is there is a case of direct evidence, not just a correlation, but evidence of oncodriver. Yeah. So, and we saw that there is activity of such compound, but then it occurs that there is also some off-target activity, and currently we are working on structure of this oncogenic mutation to continue our structure-based drug design with more specified structure, not of a wild type protein, but also with the structure of a mutant protein. And this project was supported by Bayer, but yeah. So
thank you.
Yeah, that's almost all. So yeah, we are now continue working on this. Thank you for your attention. Here there are some photos from the trips where we measured the crystals. Yeah, and this are acknowledgement to our colleagues. Thank you for your attention.
Thank you, Dr. Alexey Mishin. Very impressive work, but you have taken all the time and
yeah, sorry.
Good. So